Hello, today I'm going to go over DNA base pairing rules mini lecture, and we're going to go over the structure of DNA and how it kind of fits together, starting with the smaller pieces. So this first image here, this is a nucleotide, and this is a building block for DNA. So if DNA is like a Lego house, this would be the Lego pieces that put together that house. And we've got three parts to this nucleotide. We've got a phosphate here. Down here is a pentose sugar. And pen is going to be 5, so this is a 5-sided sugar. And then over here is just generic. This is our nitrogenous base. That's not actually what the nitrogenous bases will look like though, because there are 5 different types. We don't actually have the structure in this image of the nitrogenous base, but this is going to be where your actual genes come from, the code for your genes. This is the nitrogenous base that does that. All right, so this is our nucleotide here, our building block for DNA. So let's take a closer look at the nitrogenous bases, because these are important and we should understand the structure of those. I've got my nitrogenous bases imaged down here. Okay. Now you'll notice that this top row has double rings. So you see 1 ring, 2 rings, 1 ring, 2 ring. So those top 2 are double rings. The first one here is adenine, and the next one is guanine. Now these will not be bonding together. Adenine and guanine don't bond together. They're going to bond with the bases below them. These ones down here, you notice there's only a single ring. So these are our single ring bases. The first one in this image is thymine. And then we have cytosine. And kind of special one over here, this is uracil. We're not going to really talk much about uracil. It is involved with RNA, another topic. But when you have the double ring bases and the single ring bases, they're going to form bonds between them. So adenine and thymine will bond with each other, and guanine and cytosine will bond with each other. Guanine doesn't bond with thymine, adenine doesn't bond with cytosine. So adenine, thymine, guanine, cytosine. All right. Now let's look at the big picture. So here, we're going to start at the beginning again. We've already reviewed these. So these are the parts of our nucleotide. So we've got our phosphate. Down here it says T, so I know that is one of my bases, thymine, which makes this my sugar right here. We put all 3 of those together and we get our nucleotide. All right. The nucleotides, I said, were the building blocks. So what we're going to do is we're going to start connecting one nucleotide to another, to another, to another, to another. And when you do that, you get this long chain of nucleotides. We're going to say this is a single stranded DNA. So this is one strand of DNA. By taking those nucleotides, binding them together one at a time in a long strand, that's our single-stranded DNA. But most of the time when we find DNA in our cells, it's going to be like this here, where there's two of those strands. So this is a double-stranded DNA. And you'll notice that the sugar and the phosphate that run down each side here are not bonding across. The bonds come between the bases, right? These are the bases, and the bases bond together to keep this double-stranded DNA held together. And like I said before, you'll notice that it's A and T that bond, like down here and here, and C and G that bond. Always in that pattern. A bonds with T, C bonds with G. These were our bases, right? Adenine, thymine, cytosine, guanine, which we saw back there, okay? And then finally, when we take that double-stranded DNA and we twist it, this is our DNA double helix. And that twist is important. We're not getting into major and minor grooves, but there's something different about this double helix that when it's twisted up like this, that differentiates it from a double-stranded DNA just straight. Last couple of things here that I want to note. We talked about this backbone that had phosphates and sugars. Well, that's actually got a name. And it is the sugar phosphate backbone. This is the structural support of the DNA. All right, this is not part of your genes. That's in the bases here. Like these are where your genes are. This sugar phosphate backbone, though, holds this together, holds this double helix, gives it some structure, right? And then we've got some arrows pointing to these blue dotted lines between bases here and here. Well, those are hydrogen bonds that hold them together. Hydrogen bonds are relatively weak. This is good. That means we can break these bonds open, read the bases, and then they can come back together. So the hydrogen bonds are actually essential to be holding these together because they can open and close. Where the bonds that hold together the backbone are much stronger. They're not supposed to come apart, so they're much stronger. That's why they give structural support. Okay, I hope this helps. Thank you very much.